Hello everyone, today we'll discuss about neuron. Now what is neuron? It is a structural and functional unit of the nervous system. Nervous system consists of neurons and neuroglia. Total number of estimated neurons in human brain is about 10 raised to 12. Parts of the neuron. Neuron consists of cell body which forms the grey matter and processes they are known as neuritis. They are axon which forms white matter and dendrites. Now, structure of neuron. Neuron consists of cell body or soma or perikaryon, and it has protoplasmic processes axon and dendrites. Axon which carries information away from the cell body and it is a single process. Dendrites they carry information towards cell body and they are from 0 to multiple. Now, cell body or soma or perikaryon. It is round or stellate or pyramidal or fusiform shape. Size is variable and it contains nucleus with 1 or 2 nucleoli, and the cytoplasm contains organelle like mitochondria, Golgi apparatus, endoplasmic reticulum. It has no centriole, and absence of centriole that indicates that neuron is a highly specialized nerve cell which has lost its power of division and regeneration. And once it is degenerated, there is no regeneration and it is replaced by the neuroglia. Now, nasal granules or nasal bodies. These are the basophilic granules. They consist of ribonucleoprotein, that is RNA with protein, and they are composed of rough surface of the endoplasmic reticulum. They are important for protein synthesis. They are present in the cell body and dendrites, but they are absent in the axon. They are required for maintenance and repair, also required for the production of neurotransmitter and production of enzymes. They are also known as tigroid body, and the size in number varies with the physiological conditions of the cell. Now, chromatolysis, that is the disintegration of nasal bodies into fine dust-like particles, and that occurs in fatigue, poison, nerve degeneration and injury to the neuron. Next structure is neurofibrillae. They are fine thread-like structure with a diameter of 6 to 10 nanometer and variable length. They are made up of microtubules and microfilaments, and they transverse the soma, dendrites, and they are also present in axon. In various degenerative diseases like Alzheimer's disease, neurofilaments, protein, they get altered and they form the neurofibrillary tangles. Certain pigment granules are present in some neurons, like neuromelanin is present in the substantia nigra. And with aging, lipofuscin pigment is present in the neuron. Now, dendrites are shorter than axon. They are 5 to 7 processes. They extend out from the cell body and they arborize extensively after they leave the cell. They extend out from the cell body and their numbers may vary from 0 to multiple. They look like thorny projections because they have a spine on their surface. You can see here in the diagram, and these spines, they are receptive processes and they are the site of graded potentials which is non-propagated and summated. Now, here dendrites, they do not produce action potential but they conduct the graded potential. These graded potentials, they are summated and produce action potential in the axon. Dendrites, they have nasal granules as well as neurofibrils both. Now, axon, which is a single process, that is the cytoplasmic extension, and they have the function of conduction of impulses away from the cell body. The length varies from few micron to 90 centimeter or maximum 1 meter. Now, what is axon hillock? You can see in the diagram here. This one is the axon hillock, this one. It is the part from which axon starts. It is the cortical extension of the cell body from which axon starts. Next is exoplasm, that is the cytoplasm present in the axon. And next, that is exolemma, which is the cell membrane of the axon. Now, you can see here Schwann cells, they wrap about 100 times around the axon and they form myelin sheath, and this myelin sheath is compacted by the protein, extracellular protein, which is known as protein 0. When there is mutation of protein 0, that results in defective myelination and it also affects the conduction. Now, initial segment. What is initial segment? You can see here. This one is the initial segment, this part here. Initial segment, it is the part of axon between axon hillock and beginning of the myelin sheath. You can see here, this is the part here, small one, I could not exactly locate this one. between the axon hillock and the origin of myelin sheath. Now, another terminology is telodendria. Now, here these are the telodendria. So, these are the terminal branches of axon, which are known as telodendria. At the endings of the telodendria, there is bulb-like projection, and these are known as terminal buttons or synaptic node. Nervous system. Myelin sheath in the peripheral nervous system is formed by the Schwann cell, but these Schwann cells are absent in the central nervous system, and therefore in the central nervous system, myelin sheath is formed by oligodendrocytes. Now, myelin sheath that is formed by each Schwann cell that covers only a short segment of axon, and it is the interval of about 1 millimeter distance, and between these two Schwann cells there is a gap which is about 1 micrometer, that is known as node of Ranvier. Now, internal structure of axon. Axon has got, as we have discussed, cytoplasm which is known as exoplasm, and it contains mitochondria, neurofibrils and exoplasmic vesicles. Nasal granules are absent here. Protein required for the growth of the nerve that is transmitted by the exoplasmic flow. Next is exolemma, as we have discussed. It is the cell membrane of the axon and which is surrounded by myelin sheath if the fiber is myelinated. Now you can see here in the non-myelinated fiber, Schwann cell, this one you can see, these are unmyelinated or non-myelinated fiber. Single Schwann cell, it covers multiple nerve fibers, you can see here. Whereas in myelinated nerve fiber, the Schwann cells, they form myelin sheath, you can see. Functions of neuron. Important function of neuron is to receive as well as transmit the signal from one neuron to another. Also, nerve cell body, it carries genetic information for the protein synthesis. It has lost its capacity of cell division, so no information for mitosis. Neuron maintains structural and functional integrity of the axon by anterograde and retrograde transport. Now, what is this anterograde transport? Here, transport of substances you can see from the cell body towards the axon, that is anterograde. You can see here in this direction. Materials which are required for the integrity of axon are transported by anterograde transport, and rate of the anterograde transport is 400 mm per day. And retrograde transport, that is the transport of substances in the opposite direction from axon towards the cell body. Here the speed is half, 200 mm per day. Certain substances, they are transported by retrograde transport, like nerve growth factor or certain toxins or viruses, they are transported by retrograde transport. Now, dendrites, they are the receptive processes. They receive information. They are the site for non-propagated, graded and local potential. And these potentials, they are summated and produces action potential in the axon. Synaptic node, it is the site for the release of the neurotransmitter which is required for the transmission of action potential. Now, organization of nerve. You can see here how the nerve is formed. Nerve is made up of many bundles of the axon. You can see here there are various bundles. These are okay. Now, so this bundle is known as fascicle. Many axons, they together form a fascicle, and many fasciculi, they together form a nerve. Now, these fascicles and axons, they are covered by connective tissue covering. There are three varieties of different connective tissue coverings. One, that is endoneurium, second perineurium, third is epineurium. Now we start from outermost, epineurium, that is the outermost covering that covers the whole nerve. Perineurium, that surrounds each fascicle. You can see here. This one is epineurium, this one, outermost, this one. Then perineurium, this is, that covers one fascicle, and endoneurium, that surrounds single axon or single nerve fiber. Okay. Now, as we have discussed, myelin sheath, that is white in color, which consists of protein and lipid complexes, and that is produced by the glial cells, Schwann cell. Lipids present in the myelin sheath, they are cholesterol, lecithin and cerebroside. And as we have discussed, it is not the continuous covering. At the interval of every 1 millimeter, there is a gap of 1 micrometer, and this gap is node of Ranvier. Now, myelinogenesis is the process of formation of myelin sheath. Myelinogenesis starts at about 4th month of intrauterine life, and it is completed by 2nd year of age. And therefore, child learns most of the motor activities by the age of 2 years. Now, before myelinogenesis, you can see here, Schwann cells, they are close to the cell membrane of the axon. In this diagram you can see here. Okay, and as the myelin sheath is formed, cell membrane of the Schwann cell cover the exolemma, and the Schwann cell is present in the outermost part, and this outermost part, that is Schwann cell, that is known as neurilemma. So neurilemma is nothing but the sheath of Schwann cell. Okay. Now steps of myelinogenesis you can see here. This myelinogenesis starts with the growth of the Schwann cell. The Schwann cell membrane starts growing and it forms a multiple concentric layer around the exolemma. Now mesaxon, it is the cell membrane of the Schwann cell surrounding the axon that is connected to remaining cell membrane, that is by mesaxon. You can see here, this is the mesaxon, this part. Cell membrane of the Schwann cell which is covering, that is connected to the remaining cell membrane by mesaxon. I repeat, cell membrane of the Schwann cell which forms myelin sheath and is connected with the other cell membrane of the Schwann cell through mesaxon. Okay, and here there is a double layer, as the cell membrane is made up of lipid bilayer. So this myelin sheath is also made up of double layer of the cell membrane. Function of myelin sheath. Myelin sheath is important for the fast conduction of impulse, because myelin sheath there is saltatory conduction. Impulses or the action potential jumps from one node of Ranvier to another, and this is faster than the conduction in the non-myelinated nerve fiber. Second is insulating property. Myelin sheath acts as an insulator because it restricts the nerve impulse in the single nerve fiber. It will not allow this nerve impulse to move to the neighboring fibers. So it has insulating property. Motor neurons are highly myelinated. Why? Because myelination increases the speed of conduction, and in the motor neuron, speed of conduction required is higher, and therefore they are highly myelinated. Applied physiology, multiple sclerosis. It is a patchy destruction in the myelin sheath of the neurons in the central nervous system, and because of destruction of myelin sheath, conduction is delayed or blocked, and it is a crippling disease. So this is all about structure and function of neuron. Thank you.